Hello and welcome to Virology Journal Cafe. I will be discussing the scientific paper by Baltimore 1970 titled Viral RNA-Dependent DNA Polymerase. My name is Esperanza Lopez from the University of St. Joseph, Virology Class 2025. I hope you enjoy it. You may already know David Baltimore as the creator of the Baltimore classification system of viruses. However, he is also known for the landmark experiments that we will be discussing today. Both David Baltimore and Howard Temin, independently of one another, discovered that RNA viruses can also be inserted into the host cell's DNA. This mechanism takes place through an enzyme known as viral RNA-dependent DNA polymerase, or more commonly known as reverse transcriptase. Together with Renato Del Becco, who discovered that tumor viruses operate by incorporating their DNA into the DNA of host cells, were awarded the Nobel Prize in Physiology or Medicine in 1975. The discovery that RNA can be transferred to DNA challenged the central dogma of biology and was therefore a landmark paper that proved the existence of a special enzyme that can work in the reverse direction. At the time, the central dogma emphasized a strict flow of genetic information. DNA makes RNA and RNA makes protein. RNA viruses, however, puzzled scientists. They carried RNA genomes but established long-term infections and can cause cancer. How could an RNA-only virus integrate into the host's DNA? That was the mystery all three scientists answered in their landmark experiments, and the rest is history. Now to the Baltimore 1970 paper. The scientific background discussed in the Baltimore's 1970 paper includes infection and transformation can be inhibited by DNA synthesis inhibitors in RNA tumor viruses. The DNA synthesized by RNA viruses is genetically specific to the infecting virus. RNA polymerases occurred in virions of animal viruses. These included Vaccinia, a DNA virus, Rio, a double-stranded RNA virus, VSV, vesicular stomatitis, a single-stranded RNA virus. This information will guide Baltimore to his experiments and conclusions. The hypothesis. The study will demonstrate that an RNA-dependent DNA polymerase is present in the virions of two RNA tumor viruses, the Rausch mouse leukemia virus and the Rausch sarcoma virus. The hypothesis, if supported, will propose a new model of viral replication. To test the hypothesis, Baltimore purified retroviruses and extracted material from within the virions. He set up biochemical assays to see if these extracts could synthesize nucleic acids. By adding radioactive nucleotides, he could trace whether DNA or RNA was being produced. He also manipulated the reaction conditions, such as adding magnesium or manganese, to see how the enzyme behaved. Step number 1. Baltimore prepared a purified RMLV Rausch mouse leukemia virus and included in the purification RNA-dependent DNA polymerase, the 4-deoxyribonucleic triphosphates, magnesium ion, manganese ion, dithiothiotol for a strong stimulation, and sodium chloride for a weak stimulation. This product would be investigated for its sensitivity to various treatments. All right, so on to the experiments. For test number 1, Baltimore analyzed if RNA or DNA was in the product. The product was treated with pancreatic deoxyribonuclease or micrococcal nuclease, and degradation was observed. When treated with pancreatic ribonuclease, the product was unaffected. Now to the data. The data for the first experiment or sensitivity test demonstrates the dramatic difference in the degradation of the product by the two nucleases that show a degradation in DNA. whereas the blue arrow points to the data that shows RNA was mostly conserved. This led Baltimore to conclude that the product treated with the nucleases was indeed DNA and not RNA. To investigate whether DNA polymerase and its template were associated with the virion and not the host cell, Baltimore centrifuged an RMLV rational leukemia virus. Suspension and fractions of the gradient were assayed for DNA polymerase activity. In the graphic, we see an arrow at the top pointing to a density of 1.16 grams per cubic centimeter, in agreement with a known gradient density of the virions. Therefore, the peak shows that most of the activity was found at the position of the visible band of virions, proving that the DNA polymerase was made by the virus and not the host cell. This led Baltimore to conclude that DNA polymerase properties were indeed from the virions, or virion specific. Virions of the RNA tumor viruses contain RNA and not DNA. Therefore, the template for the virion DNA polymerase is most likely from the viral RNA. Therefore, to substantiate this, Baltimore ran a third sensitivity test on the product with ribonuclease. Sensitivity test number 3. Here, Baltimore added HTMP radioactively labeling with deoxythymidine monophosphate, which allows the incorporation into DNA and indicates DNA synthesis from viral RNA. In curve 1 and 2, DNA is synthesized. Curve 1, no treatment, DNA is synthesized. Curve 2, pre-incubation in water without ribonuclease, DNA is synthesized. In curve 3 and 4, DNA is not synthesized, or synthesized less. In curve 3, 10 micrograms of ribonuclease added to the mixture, DNA is inhibited by 80%. Curve 4, virions are pre-incubated with 10 micrograms of ribonuclease, and the mixture is added after the pre-incubation. A more extensive inhibition is observed. Less DNA is produced because the RNA template is inhibited. To ensure that ribonuclease was the inhibitor, other enzymes were added and only ribonuclease inhibited. This was evidence that RNA is the template for virion DNA polymerase. Now to sensitivity test number 4. Baltimore conducted sensitivity test number 4 to determine if RNA polymerase activity in the virions can polymerize deoxyribonucleotides when they are provided in a mixture. Now to the data. This is the data for sensitivity test number 4. Here he compared the incorporation activity of VSV, vesicular stomatitis, the single-stranded RNA virus, and RMLV, the Rauscher mouse leukemia virus, the RMLV. The VSV RNA polymerase only incorporated nucleotides at pH 7.3. At pH 8.3, the optimum for RMLV DNA polymerase, the VSV polymerase catalyzed much less ribonucleotides. Furthermore, no DNA synthesis could be detected. In contrast, the RMLV DNA polymerase incorporated only deoxyribonucleotides at its optimum pH 8.3, and at pH 7.3, the RMLV polymerase was also inactive with the ribonucleotides. The data demonstrates that RMLV virions are highly specific for deoxyribonucleotides. This led Baltimore to conclude that RMLV virions are highly specific for deoxyribonucleotides. Alas, Baltimore's last sensitivity test was the manipulation of magnesium ion, or DATP, from the reaction mixture. A preparation of the product strain of Rao sarcoma virus was assayed for DNA polymerase activity, and these are the results for the sensitivity, number 5. The results demonstrating that without magnesium ion present in the reaction, no DNA synthesis occurs. And conversely, with magnesium ion, DNA is synthesized. This is because magnesium ion is one of the cofactors essential for DNA polymerase reaction. Therefore, Baltimore concluded that RNA-dependent DNA polymerase, or reverse transcriptase, was probably a constituent of all RNA tumor viruses. In conclusion, Baltimore concluded that both the rational mouse leukemia and the Rao sarcoma virus contain DNA polymerase. The inhibition of its activity by ribonuclease suggested that the enzyme must be an RNA-dependent DNA polymerase. All RNA tumor viruses may probably have such activity. The existence of this enzyme suggests that DNA synthesis in RNA tumor virus life cycles is an early event, and DNA is a template for viral RNA synthesis. The paper ends with suggestions for further study that almost seem as prophetic as Mendelev's blank spaces in the periodic table. Here are Baltimore's final thoughts. Whether the viral DNA provirus is integrated into the host genome or remains as a free template for RNA synthesis will remain for further studies, as well as determining whether the host DNA-dependent RNA polymerase or a virus-specific enzyme catalyzes the synthesis of viral RNA from DNA. This paper was a great paper to research. I followed and stumbled my way to decipher Baltimore's simply elegant experiments in this landmark paper. I also discovered an appreciation for his technique and contributions to the subject of virology. May the Lord grant him peace. I hope you enjoyed the paper. God bless.